The Wheat School on RealAgriculture.com is brought to you by CNMC, Syngenta Canada, and the Alberta Wheat Commission.
Hey, Carrie Strauss here with RealAgriculture.com. I'm back here today with another Wheat School episode, and I've here with me Haley Catton and Laurie with Agriculture and Agri-Food Canada. How's it going today?
Hello, doing good.
Good, yeah.
Good. So you guys are currently working on a project in regards to wireworms. Do you want to tell me a bit about it?
Sure. Should I go first?
Please.
Okay, so wireworms are a big pest, insect pest of cereals on the prairies. They're not worms at all, they're actually beetles. So we have the beetles are the adult stage here, and the larvae are what look like worms. And so the wireworms, the larvae are under the ground feeding on seeds and roots and thinning crops, and are a big problem for a lot of farmers in the prairies. So being in the soil, they're very hard to access. It's been very hard to kill them with chemicals, so they've been kind of a conundrum ongoing for farmers for decades. So John and I are working on a project on trying to work with their very specific genetics and genes to try to silence the expression of some genes and try to kill the wireworms in a very specific way.
So what are you finding so far?
So right now we're just developing the early part of the technology. So it's an RNA technology, so we needed to know what RNA is expressed within the cells of our target species. And so we've collected last summer, Haley and her group collected the larvae and beetles from our target species, and then in my lab we extracted RNA, sent that out to a facility to sequence it, and that came back, and now we've assembled the sequences and have a list of all the expressed genes. And so from published literature and other groups that do RNAi, we have some favorite genes that we're looking to target right away.
So talk a bit about how you kind of determined which species and genes you were going to target.
Sure, I can take that one. So there's been a 15-year survey of wireworms on the prairies that just got published last year, and about more than 90 to 95% of them were of three species. So I have here a wireworm field guide for the prairies that was produced by myself and some colleagues, and we're focusing on three species here that were like very, very abundant. So this one, and this one, and they all have long Latin names, so I won't list them here. But that's how we determine which ones to focus on, the most abundant three. And then if we find some success, we can adapt to other species as needed. Large amount of wireworms, but the pests species are only like 5 or 6 species, at least on the prairies. There are hundreds and hundreds of species of click beetles and wireworms in Canada, maybe thousands. Right? It just so happens that 5 or 6 of those species on the prairies just happen to really like our cropping conditions and have become a pest.
So this is the first year of the study, correct?
We're starting the second year.
Okay, yeah. So any expectations going forward with the study, what you're kind of hoping to see?
So a lot of hope for this type of technology. We know the pathways exist in these organisms, and in other studies in similar organisms, so it's been demonstrated that the RNAi can work to a certain degree. So we just need to apply, some of that knowledge with these target genes that we have in mind, and to see how it works on these particular species.
So how would you see this being implemented later on? Eventually, the goal obviously is large-scale production and to get these wireworms out of the field. How do you guys see this being implemented there?
Yeah, so for our project, we're interested in a seed coat type technology, so protecting the seed in the soil and perhaps the developing seedling for a period of time.
We'd like to thank our funders, which are ARDAR, Alberta Wheat Commission, Sask Wheat, and WGRF. Yeah, they have shown a lot of excitement about this kind of work, and the collaboration that we have. We have very, very different expertises, John and I. I'm about organisms and how they react in the environment, ecology, and John is zoomed into the cell, zoomed into the nucleus of the cell.
Molecular level.
At the molecular level, yeah. I'm at the organism level. He's at an ecosystem level, and he's at the molecular level. So we're just excited and thankful that they're excited about our collaboration on this type of work.